Mic check. Okay, let's get this going. What's up, guys? This is a video for IB year number 2. Hopefully you guys are doing well wherever you are in Los Angeles or around the world. Mr. Din here to continue our lesson on evolutionary evidence. We are T-1 week away from your evolution exam. So let's get to it. I'm going to share my screen and I'm going to show you the agenda for today. It looks something like this. Pretty easy here. Number 1 is for us to finish the evolutionary evidence explanation. Number 2 is for you guys to write a written explanation of all the evidence combined. We call this a small synthesis essay. And then number 3, I have a small bonus lesson for you guys that is not part of the IB curriculum. It is a lesson on human evolution. So we talked about evolutionary evidence in general, and we combined them all to find a compelling argument about evolutionary evidence. But how does all of that apply to human evolution? So it should be pretty fun to discuss. I would love to hear what you guys think about it. That is the agenda for today. And here is where we are in our bills. We are, let's see, let's find it here. We are on page 37 of our bills. We're going to finish up today. And as you can see, the next lesson is on human evolution on page 38 and page 39. If you want, you can pause the video and write the titles down now. Again, this is a bonus lesson. And then you guys will take your exam. Stop that share. And I am going to move into the explanation once again so we can finish up. Okay, I believe last time we ended by talking about selective breeding and how the process of selective breeding is already compelling evidence to suggest that human beings can alter the evolutionary lineage of a species. And we combined this understanding with comparative embryology, vestigial organs, and homologous structures. All of that combined is pretty dang good evidence to suggest possibly that this idea of natural selection and evolution is a compelling idea. There's a lot of evidence to suggest that it is actually true. In the beginning of last video, I said there is a big difference between a conspiracy lowercase t theory and a evidence-based capital T theory. Scientists are quite careful about calling something a theory, capital T, unless there is enough evidence to support that idea, and that idea has survived the scrutiny of other experts within the field. That's what makes something a capital T theory. Well, we haven't quite finished our combined evolutionary evidence yet. So if you're inconclusive about whether evolution is or is not the truth, maybe by the end of this explanation, it'll become more clear to you. Okay, with that said, why don't we dive right in and complete what we have to complete. What I'd like to discuss now is evidence that if evolution is real, we should expect to find the remnants, the remains of other species that once existed that no longer exist. We expect to find fossils. Okay. The one fossil I'm going to highlight, we have found many, by the way, hundreds and hundreds of thousands of fossils of organisms that belonged, of bones that belong to organisms and species of the distant past. And there are a couple of organisms I'd like to highlight in our fossils here. The first organism is one that was discussed by one of your group members last semester. This is called Tiktaalik. So, man, I'm not going to try to draw Tiktaalik. It's going to be kind of weird. It is a fish that spent significant time on land? Tiktaalik. Two A's in the middle here. It is suggested through evidence found that Tiktaalik might have been what we call an intermediary species between aquatic organisms and terrestrial organisms. Tiktaalik suggests that there was an adaptation in this specific species of fish that could spend significant time on land and not dry out. One of the amazing things about terrestrial organisms is that they have the adaptation that allows them to not dry out when exposed to significant sunlight. Intermediate species between aquatic and terrestrial organisms. Then the other fossil I like to introduce you to is something we've talked about before. It's called a trilobite. A trilobite looks something like this. This is an okay drawing. Here are the bones of the legs, spine, a head as well. A trilobite is a ancient arthropod. Now, a common question here is, how do we know these organisms lived in the past? Couldn't they have couldn't they have been living in the modern day and then died and we just haven't discovered them yet? Maybe a trilobite is still living, or maybe a Tiktaalik organism is still living somewhere in the modern day, and what we found were the remains of them in the modern day. Some people will ask a question like that, and they'll ask it in good faith. Well, the answer to their question is that it depends where the bones are found. So the bones are typically found under layers of dirt and layers of rock. So these layers are called strata. And depending on the rocks and the dirt around the bones, one can use that information to test how old something is. So you use the surrounding rocks and dirt around the bones to deduce a possible age for the bones. Okay, this technique might have been introduced to you in your chemistry class. This technique is called radiometric dating. You may have heard it as carbon dating. Well, carbon dating is one example of radiometric dating, but there are other examples of radiometric dating that can be used as well. Carbon dating only goes back approximately 40,000 years, because at that point, beyond 40,000 years, there's so little carbon-14, it's hard to determine how old something is. So how do we know something is 65 million years old, which is when the peak of the dinosaurs were? That's a long time farther than just 40,000 years. Well, the way we do it is that we use radiometric dating. We date things like lead, Pb, Rubidium is pretty good too, Rb. These sorts of elements give a much older date of when rocks were formed. And so the story I recently read is pretty cool. How do we know the age of the Earth to be 4.5, 4.6 billion years old? Well, it's because the person who developed the technique of radiometric dating looked at the oldest rocks on the planet Earth and determined the oldest rocks are 4.55 billion years old. That's how we know the age of the Earth. It's a pretty cool story. That guy is named Claire Patterson. And yes, it's a guy and his name is Claire. Claire Patterson was the person who analyzed rocks to determine the age of the Earth, 4.55 billion years old. Okay, at this point, we know that there were organisms living in the past because we have the fossil record, and we know they were living far in the past because of the radiometric dating, and we know that there are some remnants of older adaptations that still belong to the modern species through vestigial organs, and we can see similarities when we compare these organisms to other species in the same phyla. Okay, but we haven't even reached the most compelling evidence yet. These two boxes are home to the most compelling evidence we have that indeed the evolutionary idea can be elevated to an evolutionary capital T theory. Okay, let's get to it. The first compelling evidence is something called comparative protein sequencing. This is exactly what the title entails. It is comparing the sequence of proteins between different species. So here's what you do. You take 5 different species, a horse, a cow, a human, a dog, and goodness, I can't even think of an elephant or something. And you find a protein that is similar in all 5 of those species. The similar protein that we're going to use is something called cytochrome C. Cytochrome C is a protein found in cellular respiration. It's found in the mitochondria. Okay. All these organisms that I just listed have mitochondria. They also have the protein cytochrome C. If you remember how proteins are made, proteins are chains of amino acids bonded together in a polymer form. Do you remember? Okay. The amino acids have a particular sequence. So we take a human, we take a dog, and in this case, take a frog. And we look at the sequence of amino acids in their respective cytochrome C. So you take cytochrome C from a human, sequence it. You take it from a dog, you sequence it. You take it from a frog, you sequence it. And in a human, the number 1 amino acid is cytosine. And then a dog, it's adenine. And then a frog is serine. Oh, sorry, did I say adenine? I mean alanine. Alanine. And then you look at the second one, and for human, you have another cytosine. And for simplicity, for human, you have yet another cytosine. Well, a dog can have this, and a frog can have phenylalanine. That's a P. See what you're doing? You are looking at the number of differences in the amino acids in that particular sequence. So you can see that in humans, dogs, and frogs, in the second position, you have 3 of the same amino acid. And keep in mind, there are thousands of amino acids that make up cytochrome C. So you have to compare them. How many differences, how many similarities between cytochrome C of a human and cytochrome C of a dog? You compare the protein cytochrome C, and then from that comparison, you can begin to deduce evolutionary lineage. Deduction. Okay, why is this so powerful? All of the evidence beforehand was done with the naked eye. You can see it. A bone here, an embryo here, this bone here, selective breeding here. You can see it with your own eyes. The difference here is that now we're moving to the molecular level. The molecules themselves is evidence for evolutionary theory. That is powerful because it's very difficult to misinterpret a molecule. A molecule is a molecule. It's got a certain shape. It's got a certain sequence. Interpretations are far wider when you're only using your eyeballs. But with actual molecules, it's very, very hard to refute. Let's get even deeper now and talk about the next one, comparative DNA sequencing. Okay, again, very similar. You're comparing the sequence of DNA. Now we're going to the root of where proteins come from. The root of where proteins come from, if you remember, is the sequence of nucleotides. So what we have here is going to the mothership. You have humans, you have chimps. Chimps is short for chimpanzees, and bonobos. And then what you do is you compare the nucleotide sequence. So let's say in the first position, you have guanine, guanine, guanine. Second position, adenine for chimps, adenine, and then thymine for bonobos. And humans, thymine in the third position, cytosine in the third position for chimpanzees, and then thymine for the third position in bonobos. And then you compare the DNA sequence. Again, DNA sequences are thousands and thousands of nucleotides long. So there's a lot of comparison to do. Comparison of DNA sequences. Okay, I cannot understate how important this molecular evidence is. This is the source of all of the instructions for a body. And if you go to the source and you compare modern species source to another modern species source, what you find is that this evidence is compatible with all other visible evidence before it. So all of this stuff that we can see with our naked eye is verified by the comparative DNA sequencing. Now, next point. This might be obvious to you, but I really want to make it clear. If two species in the modern day have more differences in their DNA, that means they are farther related apart. Their common ancestor diverged much, much earlier in history than later, okay? The modern species that have fewer differences are more similar in the modern day. So humans and bonobos are more similar to each other than humans and chimpanzees. That's how you can do the comparison. That should make intuitive sense to you. Let me say it one more time. More differences further back in evolutionary history. Fewer differences means more modern, more recent. They shared a common ancestor. Okay, I'm going to zoom out so you can see this last box. Now, this last box is a bit confusing, and I have trouble explaining it every single year for students to understand. I'm going to try it again, okay? This last source of evidence is called patterns of variation. What this is saying is that if you zoomed out, imagine you're in an airplane and you zoomed out and you're looking at a cluster of islands. What you expect to see is that farther away from a common ancestor, you see more different adaptations. So let me put this into visual form here. Again, you're in an airplane and you're looking at this island, and then this island, okay? Now, you know from radiometric dating that this island is older, this is medium, and this is newer, okay? Islands form that way. They're old islands, then new islands. Hawaii's going to get a fifth island at some point, by the way. All right. What you expect to see is that whatever species is living here has evolutionary origin potentially from this older island. And so what you see is a snake, for instance, that in our case is completely purple. This is a snake. And then in the medium island, kind of between the older and the new island, what you'll find is a snake that has a different adaptation. But it looks pretty similar to the older snake because the older snake is a common ancestor. So this snake might have half of it is purple and the other half is red, for instance. Because on that island, there's more red rocks, and so it's easier to blend in. There you go. On this newer island, you see another pattern. The snake is relatively the same size, but in this case, that snake is striped red. So this pattern of variation is seen in the geology. There is a branching outward from the older common ancestor. That is what patterns of variation means. Branching out of adaptations from common ancestor based on proximity. Proximity means closeness towards that common ancestor. So the farther away you get from a common ancestor geographically, that you would expect the adaptations to look much different. But the closer you are, then the adaptations don't look all that different. That's what patterns of variation means. And the first person to observe this observed it on the Galapagos Islands. We already talked about him, Sir Charles Darwin. So these patterns of variation are based entirely on how far away a species is from their common ancestor. And you see the variations branch away from that common ancestor. And you see it on land too. It's not just on islands. Okay, let me zoom out. Let's take a look at this. Where are we at? 26 minutes. Look at all the evidence combined. This is only 8, by the way. There are a lot more evidence to analyze. If you combine them all into a single idea and you say, interesting, I've studied this for 30 years of my life, but if I use what I learned here and try to match it up with comparative DNA sequencing, are these two ideas compatible? Do they match up well? And the answer is yeah, they match up pretty well. And if you combine all of these things together, do they match up pretty well? And the answer is yeah, they all match up pretty well. They all support the idea of evolutionary evidence, the original idea of natural selection. They all support that hypothesis. So what do you say then? You say, okay, all the evidence seems to point in that direction. What are some other ideas that can explain this? And do those other ideas have evidence behind them? So that's where the scrutiny comes in. Give it enough time, you start to realize that, man, all the other explanations for what could have happened in the past with natural selection don't seem to match up. They don't seem to have the same compelling evidence. So the one that wins out, the idea that wins out in the end, is the one with enough evidence and the one that can survive the most scrutiny. And that was Darwin's idea of natural selection. There are some biologists that say you cannot understand biology without understanding natural selection. That is really the root of all biological thought, is natural selection. And Darwin was the one to give us the idea on paper. Okay, that's evolutionary evidence. If you have any questions about it, I'm happy to discuss on Zoom this week. We do have our exam next week. I'm going to stop that share. I know I'm just beating a dead horse with this, but we are living in an age of massive misinformation. And there are a lot of problems here that one can spend years analyzing. But I think it's important for you guys to understand this point. Most Americans don't have any kind of training in scientific epistemology. And when I say scientific epistemology, I'm not talking about scientific facts. Most people don't have that either. But even the scientists themselves don't really spend a lot of time thinking about how an idea in science is elevated to truth. Okay. That's what epistemology means. Where does that knowledge come from and how is it developed? You guys, on some level, yes, you have to know the facts, but you guys, I hope you know, are given extra instruction on just how an idea is reinforced. So in the age of misinformation, someone has an idea and it's nonsense. Okay. All kinds of nonsense is out there. The earth is flat. That's nonsense. And it's because they have an idea, but they have no way of supporting it. Their methods are only to merchandise doubt, to make you doubt an established fact that the earth is indeed round. And what I'm trying to do with you guys is I'm trying to help you understand what is and is not true. Because sometimes a lie can get really, really out of hand. And that's the world we're living in now. So I guess I'm going to end with that. Miss you guys. Can't wait to see you this Thursday. And for the synthesis essay today, all you're going to do is you're going to write about the evolutionary evidence in your own words. So with that, our next lesson will be on human evolution. Hopefully you find that enjoyable as well. See ya. Peace.